Welcome to the October Offer Educational Meeting. I'm Dr. Stuart Drescher, a psychologist and a member of the board, and have been conducting these meetings, and happy to see a lot of familiar faces in the audience. This past month, I think, has been quite a month. When we were here last together, some of us were getting ready to go off on a river trip in the most unimaginable way possible. And I think after the program finishes tonight, some people may want to linger and ask them, how did you do that? But tonight we have with us Dr. Bateman to talk about the rest of the month, which was even more unbelievable. A group of distinguished researchers and clinicians from around the world gathered in Ottawa for the International Association meeting, and on the heels of that, and a smaller group of people met in Banbury, New York. So tonight we're going to have the chance to hear what's happening on the cutting edge of chronic fatigue and fibromyalgia. Dr. Bateman?
Well, originally I was to speak in November, so on schedule, and then Stuart suggested maybe this would be a good time to get together and talk about everything that's been happening relating to the conference and latest research changes, so I decided that maybe it was a good idea, so threw together some things. I'd like this to be relatively informal, if we can, because to give you a research update, this is the good news, is starting to be impossible because there's so much going on. So I'm going to talk a little bit about just touch over the top about some things that I think are important that are happening. But I'm willing to stop at any point on any one of these topics and answer a few questions as we go. So with that in mind, let's proceed. Mostly what I'm talking about today are going to be things that happened in this IACFS-ME scientific conference in Ottawa on September 22nd to 25th, which is what Stuart alluded to. The IACFS-ME, the International Association for Chronic Fatigue Syndrome, Myalgic Encephalomyelitis.org. It's the organization of scientists, clinicians, and any other interested people who want to bring together the science and move it forward. This is a small organization. It's an international organization, but it doesn't really have a very big budget. And it's a little bit like Offer, only on a more international scale, that for the most part they have a very skeleton staff, and mostly the group comes together every 2 years to bring all the scientists in the world together to present what they've been working on in the field. So that's what this conference was. It was 2 years ago. It was in Reno. And it's been 2 years, I can't believe it. And so we had it up in Canada. And there's a great tradition in Canada of following ME/CFS and fibromyalgia. They have good support groups. The Canadian definition grew up from there, both the fibromyalgia and the chronic fatigue syndrome, the CFS-ME case definitions that are considered the Canadian definition. So this conference there was an all-day patient conference, and then 2.5 days of the scientific conference. Actually, one day that was all workshops for physicians who wanted to come learn about management and diagnosis of patients. And then, let's see, that was Thursday or Friday. And then a day and a half or 2 days, I can't remember, of scientific presentations and papers and talks. The summary at the end of the conference is always given by Dr. Tony Komroff from Harvard. And the very last talk of the conference is just a wonderful distillation and summary of the highlights of the research in the last 2 years. So I'd like to encourage you, it's not put up on the website yet, but go to the ICFSME.org website and look for Dr. Komaroff's summary of the conference. And it will give you great insights about the things that were happening in the conference. I wanted to tell you, I didn't bring a printout, but I did bring some papers that have all the websites, that the web addresses that I use in this talk. And I apologize because I did this quickly as I was leaving and didn't realize the ink was running out until I was running out the door. So they get kind of faded, but I think you could probably read them. So you probably don't need to scribble them down, or you can come up afterwards if you're interested in having a list of all the websites that we talk about. The other thing I want you to know is we did purchase a DVD that Offer will have that has all the presentations. And if anybody really wanted to view any of the presentations again, maybe you could work with us to either come view them or borrow it or something so you get a chance to view some of the presentations. You can also buy it yourself if you want to. So I wanted to start, and this is very biased, but I wanted to start with what most excites me in the research right now, and that is the gene expression biomarkers that Alan and Kathy Light are working on here at the university. And I was able to represent them and present this research in Ottawa, and it was met with a lot of great enthusiasm. How many people here in the room heard Alan Light give his talk at this meeting? I just want to know how many people have heard it. Okay, so some of these things to some degree have been talked about before, but I think it still might be interesting. So I'm going to talk about sort of the research to date that we've presented in Ottawa that has been done here in Salt Lake with the team from the University of Utah, with all patients who have come to my clinic who were interested, who were willing to go volunteer to do these exercise studies. And really, the question on this research is, can we take subjective symptoms, all the symptoms we know are hard to document, and measure them in some way? Can we come up with a lab test, right? Some kind of a test to show that people have this illness, maybe lead us to making diagnoses, to evaluate severity, develop treatments, and subset patients, all of those things. And will encouraging our patients to exercise as part of the exercise protocol, sort of making the symptoms happen, will that help us measure the symptoms better with the lab test? So that's how we started this study. And the solution was to measure the expression of genes related to fatigue, muscle pain, immune dysfunction. And we know that everybody feels worse after they exercise, right? So I just want to explain a little bit about gene expression, because this is something you're going to hear about more and more in science. Gene expression is the wave of the future for trying to understand about diseases. And if you look on the right side of this, you'll see that the double helix, this is your DNA, and the DNA in your body is the template for all things that are made in your body. It's like the blueprint for every molecule, every protein, every element of the cell, everything that gets made and reproduced, everything that gets replaced. Every process that happens in your body has to start with the DNA template. And the process called transcription is when RNA comes up next to the DNA template and makes a perfect pattern of just the gene it wants to copy. And then it takes that perfect pattern away to another part in the cell where it's turned into proteins that are then turned into whatever it is that the cell wants to make. What we've learned with new technology is that you can capture this messenger RNA, it's called, and measure it. And we have templates that you can use to match with a patient's cell and see how much expression of that gene is happening in the cell. That's what DNA expression is. So if the body's responding to a stressor, let's say it has to make new receptors, something happens, receptors get used up, new receptors have to be made, you can measure that in the moment with DNA expression. This is very new technology. And it's being used across all of science to try to understand almost every disease better. And it's helping us understand many, many things. We're not quite sure how to interpret it all the time. Not exactly what we know what it means when we measure these changes in DNA expression, but that's just the start. And in our case, we're trying to measure the amount of RNA or messenger RNA that's being made from genes that have something to do with the symptoms of the disease. So far, we've looked at 48 patients a little bit more than this, but for the purposes of this study, who have chronic fatigue syndrome. About a third of them don't have much pain, just severe fatigue, and the 31%, and 69% have pain and fatigue, enough pain to also meet fibromyalgia criteria. But the key is all of these people have significant fatigue and exertion intolerance. Then we've compared it to control patients. Those are relatively normal people, not necessarily well-conditioned, but not sick. We also did a study where we looked at patients who have only fibromyalgia, and let me tell you what I mean by that, because these are all my patients. These are people who meet all the fibromyalgia criteria, but when you control the symptoms with medications, their function improves tremendously, and they can work and do the things that they need to do. For example, their pain might respond to Lyrica, and when their pain goes away, the fatigue is tremendously better. Does that make sense? And we know that there are people that no matter what you do to try to improve symptoms, the fatigue never improves, and they're kind of on a spectrum over to people who have lots of symptoms, but if you can treat them with medications, they return to a pretty normal level of function and can exercise. So I picked some fibromyalgia patients who have pain, stiffness, irritable bowel, and trouble sleeping and brain fog. We made them come off their medicines, which they hated, and do the exercise study, and those were the fibromyalgia-only patients. And then we looked at MS patients because they have fatigue, very severe fatigue, and it worsens when they're hot and worsens when they're tired and stressed, and we wanted to see what we would find. So each of these patients underwent this study where before the exercise, we drew their blood, and afterwards, after that first blood draw, they got on a bike with arms and legs and exercised for 25 minutes. So they didn't have to go increasingly fast. The rate wasn't increased. We monitored heart rate, and they just had to maintain a heart rate at 70% of maximum heart rate. Just not too fast, actually, and a lot of deconditioned people get right up to that heart rate. So they just exercised consistently, which is a challenge for 25 minutes. Everyone was able to do it. Then their blood was drawn again 30 minutes after the exercise, 8 hours, 24, and 48 hours after the exercise. And then we did some other evaluations to monitor their symptoms. And if you look on the box on the far right side, those are all the receptors that we measured. And you don't really need to know what they are, but the blue ones are receptors that work in transmission of pain and fatigue in the nervous system. They send a signal to your brain to tell your brain you're experiencing pain, or a signal to tell your brain that you're experiencing fatigue. And these are very new, some of them newly discovered. For example, the ASIC3 receptors on the top have never been studied in humans before, only in the nervous system of mice and rats. So really cutting edge technology to try to understand how the brain perceives pain and particularly fatigue. Then the sort of orangey red ones have to do with your autonomic nervous system. The second four, 5 through 8, one's kind of a dark purple. I should look up there because the colors look different on mine. But the AD2A, ADB1, ADB2, and COMT all have to do with your autonomic nervous system. And these are receptors, and receptors on the surface of cells. And we measured, the production or increase in those. And the last ones have to do with, they're called cytokines. And these are chemical messengers in the blood that change when you have inflammation or infection. So they sort of hypothesized we should be able, in one of those arenas, right, the transmission of the symptoms of pain and fatigue, the autonomic dysfunction, and then the immune signaling, we should be able to pick up something when we make people exercise, because that's when the symptoms get really severe. So these are the control patients, and the baseline level is all of the levels of those as a starting point. They might have been different numbers, but we just made 0 the starting point. And then after the exercise, which is where the arrow came, you can see there were kind of various changes in the levels of these. And this is fold increase, like a doubling, tripling, quadrupling of the number of these genes. We repeated it again 8 hours after the exercise, 24, and 48 hours after the exercise. You can see that pink one goes up, the adrenergic beta-1 receptors, and we think that we're not quite sure what that means. It may have to do with normal changes after exercise. But when you look at the patients who had CFS and FM, you can see that immediately after exercise, 30 minutes after exercise, there's dramatic increase in those genes. So something's happening, right? There's a process that's happening, and your body is making proteins very rapidly, within 30 minutes, across all those systems. So the body is responding dramatically to this period of exercise. And you can measure it again and again, a little bit different, but the same theme. 30 minutes, 8 hours, 24 and 48 hours after that one bout of exercise. And for some people, we know that the symptoms of post-exertional malaise may exist for days or weeks, right, depending on their tolerance for physical activity. And we just grouped everyone together. So we don't really know what the differences are from this slide. You can just see. And these are people I picked specifically in my clinic that I believe have an entity of chronic fatigue syndrome that's different than fibromyalgia. Now, they had pain. Some of them had pain. But I really felt like, and the thing they all had in common was exercise intolerance. All of these patients we enrolled in the study had post-exertional relapse symptoms. So that was the common denominator. Now, fibromyalgia patients experience more pain after exercise, but they don't get as sick and debilitated after exercise as what I would call chronic fatigue syndrome. Remember, there's a lot of overlap, right? So I'm just thinking about a spectrum here. So then we wondered, how does this relate to how severely ill people are? So I assigned a score to all the patients who enrolled in the trial. I called them 1 if they were severely ill, and I really felt like they could never manage living alone. They can hardly leave their house. They can't do grocery shopping. All the way up to a number 4, someone who could sustain some kind of work or school. They didn't have to do full time, but they could show up every day, be there, be consistent, at least half-time activity. That was a number 4. So you can see everybody's disabled to a degree, right? But there's a lot of variability. So then we added up all the genes at all the times for the number 1s. So this is the 8-hour, the 24, the 48-hour, all lumped together for all the number 1s. And you can see those number 1s, the really sick patients, they've got a lot of gene expression. Here's the number 2s, here's the number 3s, and here's the number 4s. So you can see that the amount of gene expression, the amount of consequence of exercise really does relate to how sick people are. And it's a little bit subjective, right? Because I just assigned these numbers based on what I knew about people from talking to them in the clinic. But sometimes I've known people for years. I have a pretty good idea, of what their level of function is. This is cool. And when you superimpose the control values, you can see this is a lot different than the normal people in terms of the average gene expression. Now, the number 4s, you can see there's not a lot of difference in gene expression. So this is a kind of gene expression that's going to really help us for the most sick people. And those little asterisks underneath tell us that it's a statistically significant difference. That means it's really a difference, but it might be not a huge difference. So this finding, I forgot I have to go back one by one. On this group right here, was about 2/3 of the patients had this dramatic expression of genes, but we discovered there was one more group that sort of surprised us. And that's, just a little delay. We pulled out the findings of just one person because I was trying to apply for disability for him. And you can see that he's the 091006. That's subject 06 compared to all the control patients. And you can see he's got a really different pattern, right, than the patients I showed you. And really the only thing he has is a dramatic decrease in the alpha-2A receptors. You see that? The adrenergic alpha-2A receptors, the purple. So we thought, geez, is that something that we should look more of? And we looked at all the patients, and guess what? About 1/3 of the patients had that dramatic decrease in the alpha-2A receptors. And all the other people, it went up. And this was just the patients with fatigue only, and here's the patients with fatigue and pain. But regardless of which category they were in, they fell into one of these two groups. About 1/3 had that decreased alpha-2A receptors, and the others, it went up. So we think we've got two different groups of people. Even that defined group, CFS, exercise intolerance, they separate into two groups. And we're thinking maybe that the group on the left are the ones with more orthostatic intolerance, the really severe POTS and orthostatic intolerance and trouble maintaining blood pressure. And that the other group may have a more complex pattern. And we have more to do to investigate that. Now this is just to look at the graphs again. On the top are the controls, and in the middle are those patients, all the patients that had the alpha-2A reduction. And on the bottom, look, those are the patients that only have fibromyalgia. Guess what? They do not even look different than the control patients. So this is not necessarily going to be a good measure of fibromyalgia pain. These tests are a measure of disabling fatigue, whether or not people have pain. We started pulling out some individual cases. I presented these to the doctors in Canada. This is one woman. All the normal people are on the left and the woman on the right. She's 24 years old. She has quite severe CFS and FM for 10 years. This happened when she was just a teenager. And she is able to maintain about 8 to 10 hours a week of income generating work because she does it at home, teaching music students. So she's really quite impaired, and you can see a pattern there. These are her scores, just for some comparison. We had the patient report how tired they were during the exercise. So on the bottom, the report of the normal people, that's physical fatigue at baseline, in the middle of exercise, immediately after exercise, 30 minutes, 8, and 24 and 48 hours after exercise. So she said, how tired are you? On a scale of 1 to 100. And you can see that the normal people, they got a little teeny bit tired while they were in the middle of their exercise, and then it went right back to normal. And all the patient groups were up high, right? As soon as they started to exercise, their fatigue level grew, whether or not they were both CFS and fibro, CFS alone, or just fibro. And you can see the big dots are the young woman I just showed you, her graph. You can see how much fatigue she had that lasted and kept going after the exercise. Here's her pain. She's a fairly high pain patient. She has really quite severe pain along with her fatigue. So she had really bad pain when she started, and it got worse from the exercise. All right, and this is another case. On the bottom are all the normal people. In the middle are all the patients with CFS and fibro. And on top is this one patient. You can see that she's the worst of all. And her gene expression just went off the map. And she's middle-aged. She has quite severe CFS FM. She's been sick for 23 years. And she's a one. Really, I don't think she could manage if she had to live by herself. She can't do her wash. She can't go to the grocery store. And just has to have someone really to help her with all those things. But she went and did this test for us. So that shows her courage to go up and get on that bike and to go have her blood drawn. A lot of these patients came back in a wheelchair on the second day and the third day to have their blood drawn. This is a 39-year-old man. He was a professional. He was a lawyer. He's been sick for a long time, but he kind of had two waves to his illness, and he was able to work part, You know how you can kind of get away with full-time, but you're not really doing full-time? So he worked for a long time that way, and then he had a relapse, and he couldn't work at all. And he is now severely ill, and you can see his pattern here compared to all the normal people. And if you look at all this data, you can see that really 4 of those genes are most critical for discerning the difference between controls and patients. The P2X4, the alpha-2A, the adrenergic beta-2, and the interleukin 10. And if you just take those 4 and compare them to the controls, you can see there's a dramatic difference. Now, this is the group where the alpha-2A goes up. So we think this combination of genes, at least in this selection of patients, could be a very clear blood test for showing that patients have CFS and that the degree of severity of their illness. How's that for cool? So the fibromyalgia patients that didn't really show this gene expression, Alan Light went back and looked at their baseline values, and he actually discovered that P2X4 and TRPV1, 2 of these gene expression markers, and the IL-10 were actually different than people at baseline before they even started to exercise. So it's possible that we can draw the blood and look at certain kinds of gene expression and diagnose fibromyalgia before people even have to exercise. One little caveat is we made them go off all their medications before they exercise. So it's possible this gene expression is going, please give me back my medicines. So I don't know what they would show if you exercise or if you drew their blood on their medications.
How long did they have to go without them?
I think just like 48 hours. So they were kind of in full-blown rebound when we went to do the exercise.
Only for CFS?
Just the fibromyalgia medications like Lyrica, And any, we didn't really exercise anybody on long-acting opioids, but the people could come off their meds to go do the study, like gabapentin or tramadol or anti-inflammatories. So the reason I show you this, because I think this is some of the most exciting research going on. We're not quite sure what it means, and it's not quite the same as your innate genes, right? The DNA. This is what the DNA is making in response to your environment, and we know that many different things can cause gene expression changes. Your baseline genetics could be different, or viruses, injuries, stressors, toxins, allergens, all kinds of things could alter the way you express this messenger RNA to be doing something, right, responding to the environment. So we hope to be able to do more to try to lead down the road. And what Alan has been doing with this large amount of data from all the patients we've sent up is looking at different combinations of genes to see if we can put a package together that separates them. And there are really some ways we might be able to do that, and I'm going to show you these graphs. If you pick, we don't have the genes on here because this is part of new science and it hasn't been published, but if you take a certain number of genes that you think most predict the fibromyalgia patients and a certain number of genes that distinguish them in the control patients, and you put it on a graph, you can see each one of those dots is a patient. So you can see that all the fibromyalgia patients have a certain number of values and the controls, but you can see that those are different lab values for all the fibromyalgia patients and the normal people. So that was 9 gene combination. You can try to predict and use it as a diagnostic test. Look at this. I didn't tell you about this. There was a group of patients who have severe depression that were about to go have ECT. That's electroconvulsive therapy. And they agreed to be part of this exercise study. So before they went to their ECT, so we knew they were severely depressed, they really had failed a lot of medicines. They went through this protocol. And you can see that they look different. Their gene expression looks different than fibromyalgia patients. Now, these fibromyalgia patients aren't really necessarily depressed. They're well compensated, they're living their life, they've had good treatment, they have a lot of fibromyalgia symptoms, but they're not depressed. So you can see that there's really only one, see the top orange dot? That's the only patient that overlapped the values of the fibromyalgia patient. So this is a pretty good discriminating combination of genes to discriminate fibromyalgia from major depressive disorder. If you look at all the fibro patients and the CFS patients, remember a lot of the 2/3 of the CFS patients meet fibro criteria, right? So they don't distinguish very well, but there's kind of a split. And if you look at all the CFS patients compared to control patients, not as good of a split. Guess what? You have to exercise them. This is measuring their genes before they even exercise, these other ones. So once you exercise them, they tend to split apart and you can see them better. So we know that that's going to have to be true in the CFS patients. And then this was looking at CFS versus major depression. And to be honest, we didn't ensure that our patients weren't depressed. And some of these CFS patients could have been depressed when they went up to undergo the study. So we're looking to see if we can use these genes, the expression of genes, to help us make a diagnosis, even across all these different types of diagnoses. Wouldn't that be fantastic if we could help distinguish depression from fibromyalgia from chronic fatigue syndrome? So we think that diagnoses are going to be possible, and we might be able to assess severity of illness. Remember how the gene expression was much bigger in quantity when patients were more severely impaired. And we think that because we looked at genes that were related to fatigue and pain and the sympathetic nervous system, we think that really supports these processes in the illness, right? That they probably are involved like we think they are, and it would be supported by gene expression. And we think this will eventually help us understand more about the basis of illness. So any questions? I wanted to highlight our research first and explain it because not everybody has had an opportunity to hear it, and probably hearing it another time makes it more clear too, because sometimes it's hard to understand. Any quick questions about that research? Oh, I want to tell you that Kathy and Alan Light are individual researchers up at the U. They're both submitted grants to the NIH. And Kathy Light, just a couple of weeks ago, was informed that she got a $1 million grant to continue to study this research. It has to span over 3 years. So it's like, $300,000 a year, and they have several employees. It sounds like a lot of money, but they're really going to be able to, do a lot more to try to understand this. All right, quick questions.
So are they doctors or?
They're PhDs.
Okay.
They are doctors with a PhD.
Will
Yes.
they be wanting more people to?
Yeah, so I better repeat the question. So the question is, will they want more subject? And the answer is yes.
Oh.
You know, if you want to, I'll just say this, if you want to be part of research, the best thing to do is call my office, call the research department, and give them your name and your information and a little bit about you, so that if, and we'll put you in our database, because we go searching through for people with certain characteristics, you know, when we're doing some kind of a study. And, you know, if you just put Lucinda Bateman, MD, in Google, the office will come up, and you can go on our website and go to the research department, and you can email them or call them on the phone, because we love volunteers. Any other questions? Yes?
I do, but I think I did call your office and no one has called me back.
So she said she called the office and no one had called her back. Well, that's possible, because there's lots going on and there's only a few of us. And we can't guarantee that we'll include you in a trial just because you call. Each study specifically outlines what kind of a case they want to be in that. And sometimes you can, if you're on certain medicines, or if you have some other illness, or if you're a certain age or whatever, you know, you don't qualify for the study. So you have to let us know you're interested, and then we'll try to work with you to see if you fit in somewhere. And then research takes a long time. We may do a flurry of activity for a week or two, and then we analyze the data for the next year, so you don't hear from us, you know, while we're working on doing the papers. Okay, so let me keep going. I'm going to leave our papers, and then I'm going to start to talk about some of the other happenings. This is, I put this here because this is another gene study, and this was a, you know, I did these late at night, and I'm sorry I didn't put the reference on here, but I can give it to you. But a woman doing research in Gulf War illness presented an amazing paper where they looked at one gene called the PON1 gene, and then they determined if the patient had two sets of that gene, two sets of a recessive gene, or a heterozygous, you know, kind of a combination. And then they matched it against all the stressors that the military people encountered, like, you know, breathing fumes and getting immunizations and taking pyridostigmine and being around insecticides. You know, there were many, many things that happened to them we think might have made them ill. And they could show that people who had a certain gene, now this is your innate gene, like what you're born with, were 8 to 10 times more likely to develop Gulf War illness if they were exposed to pesticides, or to this drug pyridostigmine, which they took so they didn't get sick if they were exposed to nerve gas. So they showed in this study that at least some people have a genetic predisposition to developing illness when they encounter environmental toxins. I think that's an interesting study. So that why maybe some people can be around it and other people can't and get sick. There was another group that had a certain gene that were much more likely to develop Gulf War illness just from hearing the warning sirens for nerve gas. So they weren't even exposed to anything, they were only exposed to the fear, right, and the anxiety of being exposed to nerve gas. So there might even be a genetic predisposition to developing chronic illness when you're put under stress, emotional stress. So I thought that was a really interesting study that may lead to more insight. There's been a lot of research on Gulf War syndrome, and it's definitely an illness. It's kind of analogous to chronic fatigue syndrome and fibromyalgia. Another interesting study at the conference was presented by, this is the father of sleep medicine in fibromyalgia, Dr. Harvey Moldavsky. And he's based in Toronto, and he has done, if you've ever heard about alpha wave intrusion in sleep studies, he's the one that did most of those really interesting studies to show many of the baseline studies. Well, he was in Toronto when the SARS epidemic occurred. And SARS, remember SARS? Stands for Severe Acute Respiratory Syndrome. Well, it was this epidemic viral infection they eventually discovered was caused by coronavirus. And many people in little clusters became severely ill. And in Toronto, this happened from one healthcare worker that flew back from China, who had been exposed to SARS, and infected all the people in the hospital that person came in contact. There were 273 confirmed cases, and most of them were healthcare workers, people who worked in the hospital. 44 people died, And they tracked, and 50 people remained disabled, 13, 1 to 3 years after recovering from this virus. And most people recovered, right? The SARS, it's a flu, right? And we know some people became severely ill and died. Well, what about the people who didn't die? So they went to those people, and they were able to find 22 of them. And guess what they had? They had post-SARS CFS-FM. They were weak and tired. They couldn't do anything without relapse. They had pain all over. They had cognitive dysfunction and couldn't sleep. What does that sound like? So this is a great case showing that that we know that CFS-FM can develop after many different viruses. But this shows a large group of people. We know exactly what they had. And they are just one example. The next thing I want to talk about wasn't really science presented at the conference. It was an announcement that happened just before the conference started. How many people here have heard about the Chronic Fatigue Initiative? Raise your hands if you've heard about it. Well, I want to tell you about it because we're going to be involved with it here. And I think it is right now the single most important effort that's been made in the field ever. This is a nonprofit that has been in the works. It's called the Chronic Fatigue Initiative. They have a website, cfinitiative.org. And they are going to do a number of projects, and I'm going to tell you what the projects are. But they are backed by $10 million to follow this through. And they're affiliated with Harvard and a number of other organizations. So the first project they're going to do is working with the Harvard School of Public Health. They're going to look and see if they can do kind of longitudinal look at people who have developed CFS. And they have a cohort of people they've followed for many decades that were nurses there or in medical school. I don't know exactly what their cohort is, but many big institutions do that. They kind of recruit medical students and they say, you're going to be part of this study for the rest of your life. And they study everything about their health from the time they're young until they die. So you have really good longitudinal, and you can look back and say, oh, all the people with high cholesterol eventually got heart attacks. You see, it makes it really easy to track. So we're going to take their big database that they've been establishing at the Harvard School of Public Health, and they're going to study CFS and see what they can learn about it. This is really important. So they have blood samples and history and everything from a group of patients they've been tracking for a very long time. And they'll see how many of them have CFS and what they think maybe led up to their getting CFS. What a great resource. The next thing they're going to do is called the CFI cohort recruitment. And this is a project where they're going to engage 4 different sites around the country that have CFS clinics. And you can see that the fatigue consultation clinic, my office is one of those. So Nancy Klimas, is in Florida, and our clinic in Utah, and Daniel Peterson in Nevada, and Jose Montoya. Dr. Montoya is the one who did the Valsite experiments. Do you remember those antiviral drug trials? And he has a CFS clinic. So we're going to enroll patients from all of these clinics, and they're going to be followed at the CFI, at this database. Hopefully at least 200 people bringing all these clinics together, all with CFS, all that are very well defined. We're going to draw their blood and put it in a biobank, and we're going to take all their information, how old they are, what their illnesses are, everything that's happened to them, everything we can think of that might give us information to understand their illness, and put it in a big computer database. And then we're going to have blood and a computer database and 200 people and 200 controls, and they're going to be able to start doing research to understand what might cause CFS. The first project they're going to do on all these samples is what they're going to call the pathogen discovery and pathogenesis study. So pathogens are the disease-causing organisms, and pathogenesis is the process, or the process of getting ill. So what happens in the body to make people ill with chronic fatigue syndrome? And they're going to utilize the talents of Ian Lipkin at the Center for Infection and Immunity at Columbia. Dr. Lipkin just did an interview on NPR about his input to the movie Contagion. Has anybody seen Contagion? So he's a virology epidemiologist, and he was in the scenes at the back advising them about how to make that a realistic movie. And he's discovered like 400 and something new viruses. So who would be better to study the blood of CFS patients than someone who has done all that unique investigative work around the world for new viruses? There's also going to be what they call the Mechanism of Illness Program. And this is going to be a grant program where they're going to continue to provide money to researchers who want to do research to understand what CFS is, biomarkers, mechanisms, and they've already picked their first grant recipient. She's going to work on the Pathogen Discovery Project. But they're going to take promising young scientists and clinicians and researchers and give them money to do research. So how's that for an incredible breakthrough? We haven't had that kind of a unified project from the federal government to this point. And so it's going to have to come maybe through this private foundation, or maybe it'll inspire the federal government to compete, to do even better. That'd be great for all of us. All right, any questions about the CFI? So we will be contacting, there was very kind of a specific type of patient they want to enroll in this particular project, and then they'll probably track this group of patients for a while, maybe a period of time. I'm not sure. We'll see. So we'll have a role in there. Okay, next thing I wanted to mention was sort of a general comment about the Centers for Disease Control. And I know that there have been a lot of maybe frustration and criticism about the CDC. I will say I think they've made an important contribution in many ways to what we know about CFS, particularly in terms of the epidemiology. But there's really a new interest at the CDC in trying to analyze data and move forward and study the illness more. And this was manifest. There were 3 papers that were presented at the conference. One was regarding perforin, which is a protein found in CD8 T cells and natural killer cells. So the CDC published a paper that sort of validated that we know there's dysfunction in natural killer cells in chronic fatigue syndrome. There were 2 other papers, one showing low aldosterone in patients with chronic fatigue syndrome. That is a hormone that helps you maintain sodium and fluids. So if you have abnormal aldosterone, that might play a role in why people have orthostatic intolerance, have trouble maintaining a good blood pressure, and having rapid heart rates of POTS. And they also published a paper this year showing that women with CFS have many more gynecologic problems, more likely to have bleeding, more likely to have procedures, more likely to have a hysterectomy, which suggests there may be a hormonal problem at the basis of that, leading to many more problems and early menopause as well. So really 3 important papers came out of an analysis of the CDC's old data. So this team is working to try to get their data out. They've also made the data available from the Wichita study. This is one of the first population-based studies. So any scientist who wants to go back and review that data and maybe do some more research can do that. And that's really important collaboration. One of the criticisms of the CDC has been that they weren't willing to collaborate with other scientists outside of the CDC. So that shows a willingness. And there's what really shows a big change to me is a project they're doing called community-based surveillance. And they are recruiting at least 6, 7, or 8 clinics. I'm not sure the number because it hasn't been fully established, all around the country who are going to enroll their patients in surveillance. And the CDC is going to learn from the clinicians in all the different sites. We're going to also be a site on that study. And they're going to study what's happened with the patients, what their other illnesses are, and learn about CFS from a much broader community perspective, which is, I think, very, very important. And they're also going to try to have a collaboration with the clinicians who've been treating patients for several decades so they can learn more and we can share what we know with them. There's also an effort called the CASA project, and CASA stands for Collection, Aggregation, Storage, and Analysis. And this is not led by the CDC, but the head of the CFS unit at the CDC, Beth Unger, is on this committee as a volunteer. And this is a group of people from the NIH, the CDC, clinicians, and researchers who are trying to come up with a standard group of tests that we all ought to use in research. And so that everyone does the same measurements. So we can all compare research projects. So this is really important collaboration and progress. Any comments, questions about the CDC? I just sort of glossed over their papers, but most of these have been published actually, and you can look up the publications if you want to read more specifics about the papers. So the next category I thought was great progress and important is the National Institutes of Health, or the NIH, have become much more proactive and interested in funding chronic fatigue syndrome and fibromyalgia research. As we just saw from Kathy Light getting that $1 million grant, and that's just one of many. But one of the pivotal things that happened this year was the State of the Knowledge Conference. How many here are aware of the CFS State of the Knowledge Conference that happened? So this was sponsored by the NIH, and people from all over the country and the nation came and presented in all the different areas of study related to chronic fatigue syndrome, so the federal government could assess what we know and what we don't know. Wow, what a great thing to do, right? Because we've been swimming with not, feeling like no one was doing research, no one's paying attention to the illness. So once the State of the Knowledge Conference has been done, it becomes a public record. And if you want to read the transcript or you want to watch the DVDs, you can go online at this website, and you can see all the science presented in that conference. And there were members of the NIH all throughout, and specialists attending in this, I can't remember if it was a 1 day or 2 day conference, it was a 2 day conference. And the director of the NIH came at the end of the conference, and the director of the Office of Women's Health, Research in Women's Health, came to the conference. So there's really very renewed interest in CFS, and not only that, they've adopted the term ME-CFS. So they are now saying ME-CFS, the ME term on it, without just the CFS alone, which shows an effort to want to integrate and collaborate with what's been done so far. Now, the Office of Women's Health, Office of Research in Women's Health, is an office in NIH, and that's the home of CFS research. And then there's also a committee called the Trans NIH ME-CFS Research Working Group. And this is a group of people, one from each of the different agencies throughout the NIH, who sit together on a committee to make sure that all the agencies across the National Institutes of Health take CFS into account when they do research and when they fund. So that is very, very important research. And if you want to go to their website, I've got the site there. So really, these are very, very important. Those of you who came in late, I have 30 copies of these websites up here. So maybe not one for everyone. I didn't know for sure how many people would come. So if you want to have a typed copy of these websites, you can. And if anybody wants a copy of my slideshow or that I didn't print out, if you would email the office or call the office, I could send one to you by email. Not everyone who hears this tape, just the people here in attendance. We might have a bigger job than we could do if we did that. All right, the next thing I want to tell you about that I think is important is what's going on at the CFIDS Association of America. How many people have been to the Research First website and looked at it? Okay, so for those of you who don't know, you can just put Research First in Google. Make sure you make it a 1, ST. Don't type out the word first. And it will take you to the website. And this is a really good website designed just to present to you the ongoing research that's really making waves right now. They also have a publication, this just came out. I don't know how regularly it comes out, but that summarizes what's going on, and you can subscribe to that and get that. But if you really want to know cutting edge, what's happening, all the controversies, you can go to this website. And there's a blog there, and you can you can participate, you can ask questions. It's a really great website for learning about the most cutting edge research right now. The CFIDS Association, I want to commend them. They took a big risk a couple of years ago. And in 2008, and I was on the board at that time, they decided to, just because they had limitations, right, of what they could do, to downsize the educational side and really focus on research. And the board said, what really needs to be done, right? We need more research. We need more research. We've got to move the field forward. So the organization decided they were going to take a risk, and we set a goal that was unbelievably unreachable for this organization at that time, to raise $1 million. Well, because of all the wonderful people out there who responded, the association did raise $1 million in donations. And that $1 million went to fund 6 projects, one of which was Alan and Kathy Light's project. And if you look at the 6 projects that they funded, it has led to at least $5 million in grants from the federal government to go on and study CFS more. So that risk they took at that time really translated into great progress for science. Because then the NIH can take over, the federal government can take over and fund. So, Kathy and Alan Light got a relatively small grant, and now Kathy has $1 million, and we're hoping Alan Light's grant request will also be funded. And then we'll really have the juices flowing for doing more research and making progress. Okay, that's enough for that. The next thing I want to talk about is XMRV. So the bottom line, this was quite amazing. Right in the middle of the conference, there was breaking news all over, at least the websites related to XMRV, that the Scientific Research Working Group, which is a group that was looking at XMRV, looked, and they sent blinded samples to a number of labs, including the labs that have been diagnosing, showing the positive XMRV. They sent them samples that had known XMRV, and they sent them samples from patients that have, so these weren't from people, they were from the lab, so known samples of XMRV, and then they sent them patient samples, and they sent them control samples. And the very unfortunate thing is, when it was all over, no one could diagnose XMRV. So even the labs that had been finding XMRV before were no better at finding it in the real XMRV. They found it in a certain percentage in the normal people and a certain percentage in the sick people. So very unfortunately, it demonstrated that the quality of XMRV testing right now is not good enough to ever make a diagnosis. This is very disappointing for everyone. So XMR, and so the upshot was we really can't test the blood for XMRV because we don't have a test. And if you actually think about it, the test that's used is the test that we've used for everything related to XMRV. All the research, everything we've done so far is based on tests that probably are unable to reliably measure XMRV. So this is a very big disappointment for everyone involved in the field. So XMRV is a real virus. It does exist in mice. It doesn't infect them. And there really, so far, is no convincing evidence that it causes disease in humans. And that's the current level of thinking of XMRV. Now, there is still a study that's supposed to be funded by the federal government to look for XMRV, and we're one of the sites that's going to provide patients for that study. And I can tell you honestly, I'm not sure. As far as I know, we're still going to go ahead with that study to see one more very big study.
Attention. An emergency has been reported in this building. Please cease operations and leave the building utilizing the nearest exit or fire exit stairway. Do not use elevators. Repeat, do not use elevators.
Well, I guess we have to go out.
Attention. Attention. An emergency has been reported in this building.
Well, let's see what happens. But
Cease operations and leave the building
okay.
utilizing the nearest exit or fire exit stairway. Do not use elevators. Repeat, do not use elevators. Attention. Attention. An emergency has been reported in this building. Please cease operations and leave the building utilizing the nearest exit or fire exit stairway. Do not use elevators. Repeat, do not use elevators.
Kind of far. As we always used to call them as kids anyway.
Okay. I'm going to keep going.
Can I ask a question on that?
Yes.
Have they disproved the involvement with
I can't speak with certainty. The question is, have they disproved the association of XMRV with prostate cancer? And I would say it puts all of that research in question. Definitely. Now, each one of those labs has developed their own assay, so the comparability is not perfect, but it's not very encouraging. Yeah, it's very disappointing. Any other questions about XMRV? Yes.
How is the I guess it's not Peterson Medical Institute, but the original institute, how are they responding to this?
You know, you might want to, the question is, how has the WPI, the Whittemore Peterson Institute, responded to this? And they've responded the best they can. They've kind of rolled with the punches. If I were you, I'd get on the internet and read. There have been massive publications in the last week. And Dr. Mikovits is no longer working there at the WPI as of this week, over other issues, apparently, released by the Institute. And there's been a retraction of elements of the paper in Science, and there's now raging controversy around it. I think I'm sort of the calm type. I look at this as, we need to take the emotional part out of it and look at it as science. Very, very often in science, good ideas do not pan out. So you must just forge forward, right? When we subject our studies, our biomarker studies that Alan Light and Kathy Light are working on, to replication and wider group of patients, I have no idea if they'll come up with the exact same things we did. They may not look at the same patients. There may be many issues, and we really can't get too excited about our study until it is, been replicated and everyone can do it and it's reliable, and that's true with any studies for viruses and particularly for XMRV. So I feel horrible for all the researchers who've built their whole lab around it, who've done publications and had graduate students working on projects based on what may be, by no one's fault, a contaminant in all of these reagents that's been there for many years. And until our technology got fine enough to detect it, we didn't even know it was there. And so all this research got based on it. So I like to take the emotion out of it and just look at the facts, and we back up and we go another direction. And whatever the truth is, it will eventually come out. And there may be some destruction in the wake, but hopefully as little as possible. Hopefully people can redirect and put their energies back into things that are going to continue to have promise. Any other XMRV questions? Okay. All right, a few more points for the conference. I thought this was an important point. There was a debate at the conference between two very prominent fibromyalgia researchers, Dr. Daniel Klaw and Dr. Roland Stodd. And they've both done, dozens and dozens of papers and studied tender points and studied fibromyalgia. And they did a counterpoint of, should we use tender point criteria or should we throw tender points away? The tender point exam is what we do to make a diagnosis of fibromyalgia. And you have to have at least 11 of 18 tender points to meet the fibromyalgia criteria. The funny thing is when this debate got started, they realized that both of them kind of felt the same way. That there used to be some utility for tender points, but there's probably very less and less reason to require tender points. And there's also there are new fibromyalgia diagnostic criteria that you may be aware of. These aren't brand new, that have been published, but they haven't been widely adapted yet. And in order to diagnose fibromyalgia by these new criteria, you have to, they assess these areas. Widespread pain index, that's the number of areas in your body that are painful. You can get up to 19 points for that. A score for fatigue, a score for sleep, a score for cognition, and then one more score for kind of add up all the other million symptoms and put them in one bucket. And if there's a lot of them, you're a 3, and if there's a few of them, you're a 0. Then you add that score up. And if your score is high enough, you meet fibromyalgia criteria, but there's no tender point exam involved. My thoughts about all these evolving definitions, it's frustrating because we keep broadening the CFS case definition and we keep broadening the fibro case definition, and we're really just going to be defining one big group by the time we finish, which makes it difficult for us to tease apart the subsets. But the way I look at it right now is maybe it's good. First we'll define the group and put everybody in a pot, and then we'll look at subsets. Because we need to validate everyone. We need to validate that the illnesses are real. And I think the fibromyalgia researchers and the chronic fatigue syndrome researchers need to talk and crosstalk, and we need to include the criteria of each in all of our research. And until we do that, we're not going to make sense out of this. So hopefully down the road it will start to make sense. But you may see tender points disappear. And I don't think anybody will feel bad when they don't get pushed on in all their tender points. Anybody wants to keep the tender point exam? We'll bring you up and I'll demonstrate one. All right. Another big bombshell that happened around the time of this conference was a paper that was published that was a revision of the Canadian consensus criteria for ME/CFS by Dr. Crothers, who was the lead author. Now, you may be familiar with the Canadian case definition for ME/CFS, and really the intent was to revise it and improve it and put more footnotes about the research that supports it. But it was published with a new name as well as the International Consensus Criteria for ME. And really, one of the purposes of this group is to get rid of CFS, the name CFS. So not only did they drop the term CFS, they dropped fatigue as a criteria for the illness. The primary criteria is post-exertional neuroimmune exhaustion, or the acronym is PENE, P-E-N-E. So the hallmark symptom of disease is intolerance for activity and all the symptoms related to trying to be active. We all understand that, right? And then the other criteria fall in those symptom clusters. Well, I thought this wouldn't even make a wave. There were many of us in the community that added input to this paper and ended up as co-authors on the paper, but it's created major controversy as one more case definition. So it's really ruffled the waters, and it's gotten everyone in the field talking about how we should define a case. How do we define a case of chronic fatigue syndrome? How do we define a case of fibromyalgia? I think discussion is good. I think the more we talk, the better we'll be as we go. Any questions about that before we go on? Okay, the last ones are a little quicker. Yes?
What is ME?
ME is myalgic encephalomyelitis. So ME is the abbreviation for that long word, and it is the name used outside of the U.S. for severe debilitating chronic fatigue syndrome. Now, there's some debate among all these groups about what each defines. But in the U.S., we've tried to just adopt that term because it's a term that is more acceptable and is a little more descriptive than chronic fatigue syndrome. And unfortunately, chronic fatigue syndrome has come to be a term that is thrown around and misunderstood and stereotyped. And so many people have attitude about that before they even can understand what the illness is. We all understand that. I think if that had never happened, I don't think the name would matter. I think if we had a good diagnostic test and we could show that people were sick, it wouldn't matter what we called it. But because we don't, and people can stereotype and generalize and make the wrong conclusions, it's become a very difficult tag name. And also you get all those comments like, oh, you're tired, I'm tired too, and all those things. So ME is the shortened version of myalgic encephalomyelitis, and you can see why we say ME instead of that mouthful. And we're tagging it onto CFS, and I think the idea is to call it ME/CFS and pretty soon just kind of drop the CFS. And that's what that next published case definition was hoping to do. Just move to ME and get rid of that old term. There's a lot of conflict in the field about, in changing the name, what ought to be the right name, and I'm not going to take sides particularly. I have my own opinions. All right, I just wanted to highlight a few other studies I thought were important at the conference. One was, I think you probably remember that the University of the Pacific group published data a few years ago showing that if you did cardiopulmonary testing on day 1 and you did it again on day 2, that people with CFS could not replicate their test. There was something metabolically wrong with them. And it really was sort of known in the CFS literature, but had never been replicated. So this is a different group who also brought a number of cardiopulmonary tests. They did cardiopulmonary testing on a research basis, and they could show that on day 2, the VO2 went down, that's the oxygen consumption, that the anaerobic threshold went down, and the work at anaerobic threshold went down. So even though a patient worked just as hard, they could not produce as much energy. They really had a different metabolism on the second day. So we think this is showing that when post-exertional malaise is present, they're more sick and less able to function. So this is really important confirmatory test that hopefully will continue to move the field forward as we look for objective measures to use for disability, to use for making a diagnosis. This was a study I thought was meaningful. They're all meaningful, but especially meaningful because I'm really interested in the young people who get CFS. What happens to all those kids who start as a teenager or get sick in their 20s? And Catherine Rowe is a CFS specialist, a pediatrician in Australia, and she looked at almost 800 cases of CFS, of kids who are 6 to 18-year-olds when they became ill. And they're with the primary symptom of exercise intolerance. So we're trying to distinguish them maybe from other kinds of fatigue. These are kids who really can't exercise. They were able to interview 342 quite a while after their illness, somewhere between 2 and 21 years after they got ill, to see what happened to them. You know, how many got better, how many went on to stay ill. And of the people they looked at, the average length of illness was 5 years, but it ranged from, actually I don't know if that I'm going to have to back up and say I'm not sure if the 1.7 to 21 was how long they'd been sick or how long it was when they did the follow-up. So I'll have to go back and look at that. Anyway, 60% were better at the 5-year mark. That's really hopeful. They'd recovered. And 88%, 12 years after diagnosis, said they were better. That's a long time, but it's better than never getting better. But they still had to be careful. So even though they said they were recovered, actually they were being careful not to relapse. But that's good. It's still better than nothing, right? 90% of those kids went on to finish school and got their education, and many of them got married and had families. So I think this was a really kind of positive report. It's tangible. It's helpful. I have a lot of parents who come in and say, what's going to happen to my kid? And this gives us a little bit of structure. It's not really good news, but it's at least answers to what might happen and that you should persevere. And the things that help people manage the best were balancing their social, physical, and educational pursuits. Trying to have a balanced life, even though you have to pare it down. The people who did the best were really people who tried to balance. Any questions or comments about the pediatric population? In contrast, Lenny Jason looked at adults with CFS, and this came from the Chicago epidemiologic study that was done back in the mid-90s. So there were 213 people with CFS who were identified back in the mid-90s. 10 years later, they went back to see how they were doing. And they, some of them got worse, some got better, some went from partial fatigue to full fatigue, some, went, but overall, 67% were still impaired 10 years after they're first identified in there. And what was surprising is they had a much higher mortality rate than the control patients. So they're just sicker people who had more problems. So that the adult data I don't think are quite as promising. And I think we feel that way, that the kids tend to get better, adapt to their illness, or have a resolution of their illness better than adults. This was a survey done by the CDC to see how attitudes have changed about CFS. And I think these are revealing and helpful. So 95% of healthcare providers said they'd heard of chronic fatigue syndrome. That's good. You would hope so. What about those other 5%? Who the heck are they, right? Well, it used to be worse. 71% ranked of healthcare providers said that they would say CFS is both medical and psychiatric. We'll come back to that. And only 30% of the public said that. When they were asked, do you think this is completely psychiatric, only 2% of the public said yes. How's that for progress? Very cool. And I think some of that relates to the public awareness campaign. The CDC and the CVIDS Association, spent a lot of money on public awareness for a period of years. And I think you can see it right there. Only 2% of the public said they thought it was a psychiatric illness. 14% of healthcare providers. But I want to go back up to that joint medical and psychiatric and say, when people interface with doctors, by the time you get to a doctor who diagnoses you, many times you also have depression and anxiety, and your life is a mess, and you feel like you're falling apart. So doctors really do see patients with both, the medical causes and the psychiatric causes. What they need to do is follow people a long time to adapt to their illness, to understand how to separate those out when they're making a diagnosis. But they're greenies, they're kind of new to the field. So the public were more likely to see CFS as a medical condition. And then they asked the members of the public, if you want to learn about CFS, where would you go for information? And 72% said they'd go to their family doctor. So you can see it's very, very, very important that we train family doctors about these illnesses, because that's where people are going to go when they need information. The next best is the internet, right? We know that, you can get anything what you want from the internet. Not too many people would trust a specialist, and support groups fell really low because they don't understand, what the quality of information would be when they go to that kind of a source. A researcher from Australia brought many papers that I think validate and confirm what we've known about immune testing, about natural killer cell function being reduced, and other studies related to the immune function. And I threw this one in. John Chia is a researcher in California who's been studying patients who get CFS from enterovirus infection. And he's been using a Chinese medicine. There's no treatment for enterovirus. And he's Asian. He went back to his Asian heritage and drew from traditional Chinese medicine, has been using a Chinese herbal immune booster. And he believes that his patients, he proves they have enterovirus. He's an infectious disease specialist. He treats them with Chinese medicine and they improve. And he presented a paper at the conference that if you combine it with rifampin, which is an old-fashioned antibiotic, that the patients become very ill. It's like they get a viral release and a flare, and then many of those patients go on to do much better. So I thought that was interesting, and maybe I'll try some of my patients on the Chinese medicine that he did in his study. All right, well, that's the end of my slides. We don't have too much time left, but I'm sure we might want to take 10 minutes or something to ask a couple of questions. So does anybody have any questions? Yes?
Actually, you say a lot of doctors don't know. Is it, I guess I can ask it after one-on-one. Because I cannot find I was diagnosed with fibromyalgia. The doctor that diagnosed me doesn't know.
Right. So her question is, those numbers seem better than reality about the number of doctors. I think doctors recognize that the illnesses exist pretty globally, and many more will make the diagnosis. A lot of people are a little uncomfortable making the diagnosis because they don't feel like they know enough. And then they really aren't sure how to treat it. They get very uncomfortable. I talk to family doctors and internists. I probably talked to several hundred primary care doctors in the last 3 years. One-on-one, office situations, dinner talks, from going and talking about the fibromyalgia drugs. And they're interested. They do want to know. But they haven't been taught how to manage it in their training programs. And they're kind of discouraged from taking all the information from pharmaceutical companies. And that's where most of our really good information about fibromyalgia has come from in the last few years. All the advertising you see, all the education for physicians really been paid for by pharmaceutical companies. And doctors are trained to look at it suspiciously and really, get their training elsewhere, but it isn't very common anywhere else. Starting to be. So little by little, they're going to get some confidence. But it takes a long time to learn how to treat a complex illness. So I feel like it's the duty of every patient to train their physician. So you pick one physician, work with them, tell them about your symptoms, talk to them about how you respond to medications, bring them information, and you will, as a case of one, increase their awareness of the illness. And little by little, more and more physicians will be better.
What about at the university level? Are they training? Are they teaching? They haven't taught CFS.
So the question is, what about at the university level? And I would say that academic institutions are the most conservative and the slowest to adapt new science. I wrote a little blog on this on the CFIDS at the Research First site, so if you want to go read my opinions about that big gap between, the powers that be and our clinical world. But I think it will slowly change. And to the credit of our institution here, I have probably, between me and my PA, Patty Jays, we've probably given a dozen lectures to nurse practitioners, PA students in training. They're a little more open to it than the medical training. And getting into the curriculum of medical training requires a lot of evidence-based medicine. So when the science comes, the education will follow. That's why we have to invest in science. And I know it seems like forever, but it's still about biomarkers and science and proving the illness. And when that happens, it will all follow very quickly.
How far out do you think we are from having the lights study duplicated by somebody else?
So the question is, how far out are we from having the data that the lights have done be duplicated or replicated? They are in the process of trying to gear up to do a much larger study and maybe to take their technology and have other sites apply it in satellite in a research mode. It hasn't been planned yet, so I can't really explain it. But they do have kind of an idea of where they want to go to replicate and fine-tune their science in larger populations. How long that will take? More than years, before it really becomes like, a diagnostic test, if it does come to that. But, you never know. Depends on a lot of factors. When there's a breakthrough that is real, and if it's readily replicated, it might move very quickly. If it's complicated and, maybe I select patients in a different way than a big old academic community clinic would, and they can't quite see how to make it fit, it might take longer.
Is there an interest?
Is there an interest? There is an interest. There's an interest. The federal government shown an interest because of that grant, right? They really want them to move it forward, and there's some commercial interest too in helping them raise money and do more research. So yeah, I think it's promising.
What is the difference between chronic fatigue syndrome and fibromyalgia? I heard that with fibro you have pain and fatigue, where with chronic fatigue you just have the fatigue. Is that?
I'll do my best to answer that question. So that's a big question.
Sorry.
The question is, what's the difference between chronic fatigue syndrome and fibromyalgia? First, I'm going to say no one can agree on that, even in the scientific community and the clinical community. So I'm going to explain to you what I think the current state is. Think of these diagnoses as merely a description of a collection of symptoms. So fibromyalgia is defined only by the presence of widespread pain and tenderness. It does not exclude anything. So if you have widespread pain and tenderness, that means you're tender all over, and the tender point criteria are met. That is fibro. We defined it. It isn't really a disease, right? It's a way of capturing people with similar symptoms and putting them in a group and studying them. So we've kind of come around to saying this is a syndrome. It is a syndrome. A syndrome means it's a set of symptoms we see occurring frequently, but there could be many paths to getting those symptoms. It might be multiple causes. We see fibromyalgia occurring in the presence of many other illnesses. So fibromyalgia is not really one disease. It's really a description of the presence of, the way we're defining it currently is these upregulated pain pathways in the brain. Chronic fatigue syndrome, on the other hand, is similar. We say if you have all these symptoms, we're going to say you have chronic fatigue syndrome. So if you have fatigue that debilitates you, and it's not explained by anything else, and it impairs your ability to function, and if you have post-exertional relapse symptoms, post-exertional malaise, you try to do things and you get sicker, and then you just have to meet 4 of the 8 symptom criteria. Actually, post-exertional malaise is one of them. And that's, headaches, myalgia, arthralgia, unrefreshing sleep, cognitive dysfunction, sore throat, and tender lymph nodes. So various presentations can be plugged into that definition. The thing they all have in common is they have no other diagnosis, right? Which makes it important to group everyone together. And they may have pain and may not, right? Because you don't have to have all 8 of those criteria. And if you don't have the pain criteria, you still meet criteria. Many people with fibromyalgia syndrome also meet the CFS case definition, because if they're tired and don't function at the same level, they meet those criteria, because the fibromyalgia criteria all fall in those 8 criteria: headaches, myalgia, arthralgia, cognitive dysfunction, fatigue, and disturbed sleep. So they're not mutually exclusive definitions. Now, definitions are different than people, right? And there's a broad array of disease states out there that fall under these definitions. And there are people who are severely ill and impaired with no pain, and some with horrible pain. And on the other end of the spectrum are people with all this stiffness and pain and fatigue, but the medicines help them, and if they exercise, they actually gradually get quite a bit better. That's what I would call fibromyalgia. And on the other end of the spectrum, people who can't exercise at all. So there's many different ways of grouping people. So I wouldn't consider the case definitions to be a disease that's exclusive. Does that help? So we should just be a little bit loose, right, about the way we use these terms. And when you meet the criteria, it puts you in the group. But we need to do research to define subgroups. For example, like we did in our study, we want to study only people who can't exercise without becoming much more ill. We're going to look at that group. Doesn't mean they're better. It's just a place to start and see if you can come up with some defining tests. We also have tests for people who have those fibro symptoms, people who can exercise and do better, but they're stiff all over and they have amplified pain. And we know drugs work better for them. They work a lot better for those people's pain than the CFS people's pain, the people who can't exercise. So we're learning. We're starting to subset. So I think it's best to just be kind of easy and use it as a working diagnosis, and then you apply treatment and see where you sort out. And that's really the way we approach treatment. Yes?
Are there other medications for fibromyalgia? We've heard of Cymbalta and Lyrica.
There are 3 approved medicines, Cymbalta, Savella, and Lyrica. And Savella and Cymbalta are antidepressants that raise norepinephrine and serotonin. And Lyrica is an anticonvulsant and anti-seizure medicine that alters pain pathways. And its cousin is gabapentin or Neurontin. But it went generic before they could do any studies to get it FDA approved.
Do you find that either one of them work better?
They all work somewhat in some people. So you can't really rank them because everybody's different. Some people respond great to one or the other or don't tolerate it, and some people are on both and they're doing great, and some people can't take either one. So.
I had perhaps Cymbalta, but every side effect that it mentioned, no benefits. Tried Lyrica, and so far it's been very easy.
So there's an example. So I would just caution everyone, don't base your decisions on any one person's experience with any of these drugs, because there's a broad spectrum of the way people respond. So they're tools, and you work with your doctor to try them out and see what might work in your situation, and then continue to work on non-drug interventions too, to see if you can improve your sleep hygiene and do whatever exercise you can do given where you are, and all those things until you get as better as you can get. Yes, one more.
The brain fog, the cognitive stuff is really bothering me. Are they studying why that happens, or what can a person do to try to manage that?
So the question is, what do you do about brain fog, and what do we know about it? And I will just tell you, there's not an easy answer to that question. There's probably many contributors to brain fog. If you don't sleep, you get brain fog. If you take medicines, you get brain fog. If you're depressed or anxious, you get brain fog. If you have too much pain, you have brain fog. When you're tired, you get brain fog. We think there's some studies that show that there's elevated lactate in the brain in people with CFS. I can't speak about the fibro end. And lactate is made in certain situations of abnormal metabolism, or where the cells aren't oxygenated, or many other situations. We have a lot of studies that show there are regional blood flow abnormalities, so the brain doesn't get well perfused all over in patients, and that's when they're lying down. What happens when they stand up? So we know there are some things we are looking at, but we haven't teased apart all the parts of it yet. I really say for brain fog, think of it as mental fatigue instead of physical fatigue, and just do the same thing for your brain that you do for your body. Which is get sleep and make sure it has glucose, and don't overstress it, and watch out about too many medicines because they can add on to brain fog.
Oxygen flows brain.
And yes, make sure your brain has oxygen. That's good. Don't hold your breath too long. If you've got sleep apnea, treat it. So I think we better end. We can really never answer all the questions about how to treat this and diagnose it, but I really wanted to just deliver with some enthusiasm that the science is growing. There are many more people involved. There are many more avenues of research. Research is being replicated, and I think we're picking up steam. So be a little hopeful and get engaged somehow. And if you want any of those references, come on up and take one of these papers. Thanks.